So it's a distinct pleasure to introduce our colleague and a friend, Steve Zeikner, here at UVA. He's a professor in the Department of Pediatrics and Microbiology, Immunology, and Cancer Biology. He obtained his PhD in microbiology from University of Chicago and spent a number of years as a principal investigator in the NIH intramural program, and then moved to George Washington University in the Children's National Medical Center. He's going to talk a lot about vaccines, which is what I requested Steve to talk about, because a lot of interest in the group, and he has done some remarkable work as well over the last few years. In particular, I'm aware of his recent work with our colleague and friend, XJ Meng. I think Steve is going to cover that on their plans and their efforts to develop universal vaccines for a host of different diseases. So Steve, thank you very, very much for talking to us today. This is a very broad audience, folks who do work in mathematics and epidemiology and computing and AI associated with it. We also have a lot of external folks who have joined us as well. We're part of the program. Anuj, Simon, I can see is here, and others. So you have a very, very nice audience. So take it away, Steve. Thanks.
Okay, well, thank you, Madhav, and thank you everybody else for the kind invitation. I should start out actually by congratulating you and everybody there and your recent success in getting the P2P money. I think that really opens up a huge number of very exciting opportunities, and I look forward to seeing how that develops. I'll also just first place the seed that I think things that I'm going to talk about could enable some very interesting collaborations between people, between me, my lab, and members of the audience and other people in the Biocomplexity Institute. I'll try to plant the seed for that in the beginning, and then at the end, be a little more explicit in what I mean. But if you let it percolate around in your imagination, I think you can maybe start to think of some things. I often talk to clinical audiences, and I put this automatic disclosure thing up. LVG has filed provisional patent applications for some of these things. And of course, I could get money from that, as you know. So I'm going to talk about vaccine development and new technology that we're working on. And none of the things that I'm talking about is in any way meant to disparage the amazing achievements as far as mRNA vaccines for COVID. I got my booster on last Friday, and I'm going to spend this afternoon working to vaccinate kids in the battle building. It's a remarkable achievement, but I will say that effort was really more driven by the technology. In other words, let's use mRNA, try to make vaccines out of it, and see how far we can push it. I think what we're trying to do is a more, I would say, traditional engineering approach where we try to define some end desired characteristics of a vaccine and then find enabling technologies and approaches to let us get there. And so I think if you're looking for a vaccine that would be particularly appropriate in a global context for pandemic or bioterrorism responses, in addition to having something being safe and effective, it's critical that it be inexpensive, easy to manufacture at volume, using inexpensive and readily available feedstocks, and ideally produced in existing factories available in many countries around the world right now. And of course, easy to store, transport, and administer, ideally non-parenterally. So the technology that we came up with to address those goals is expressing vaccines on the surfaces of inactivated genome-reduced bacteria. This is a synthetic biology-based approach. And for those of you who aren't wet lab people, we can now order from a dozen different companies around the world DNAs at a cost of probably less than 15 cents a base pair right now. The technology that we're employing is highly established, greater than 100-year-old technology, killed whole cell bacterial vaccines. That means that the bacteria we can just grow up, induce expression, and then kill them, which is extremely inexpensive. We present the antigen on the surface of the bacteria, which means that in some sense, the whole bacterial cell could serve as an adjuvant to enhance immunogenicity It's easy to engineer bacterial vectors further to enhance expression, antigenicity, and so on. The mechanism that we're using to place these proteins on the surface of the bacteria has chaperonin-like activity, which can promote the correct folding of the protein antigens that we're trying to make. Some viral envelope proteins, which are the targets for many vaccine development efforts, are bound by antibodies that not only interact with that envelope protein, but in the case of an envelope virus, also interact with the viral lipid bilayer, which means that presenting an antigen on the surface of a bacteria in the context of a lipid bilayer may be helpful. And the killed whole cell bacterial vaccines have been used in non-parenteral immunization approaches. And I think right now the best example of that is the WHO approved and pre-qualified cholera vaccines that have been stockpiled around the world to deal with potential cholera outbreaks. Are killed whole cell bacterial vaccines reasonable? Well, ever since the era of Pasteur, the paradigm isolate, inactivate, and inject has been applied to these killed whole cell bacterial vaccines. Here's a photo of a British soldier receiving a typhoid vaccine, of course, more than 100 years ago. And there are a number of people who originally were from India here, and you will know that you have a national hero, Vladimir Hafkine, and his institute, and he developed the early plague killed whole cell vaccine. Right now, I mentioned there are killed whole cell cholera vaccines that are pre-qualified by the WHO and stockpiled around the world. These vaccines were developed by the WHO International Vaccine Institute in Seoul, and a South Korean spin-off company from that, U Biologics, wrote a paper a couple of years ago that described their experience. And they said that from a single 100-liter fermenter, they produced 6 million doses of the WHO pre-qualified killed whole cell oral cholera vaccine at a dose of about $1 a dose. That means that were you to go and look at the fermentation tanks in Three Notch Brewery, that capacity, just to give you, I'm not saying you should make vaccines at the Three Notch Brewery, but that level, that capacity would be sufficient to provide enough vaccine for the entire US population. Cost is critical. So the current mRNA vaccines cost about $10 a dose, but that's a huge challenge when you have 7.8 billion people in the world, 2/3 of which live on less than $10 a day. It's also very interesting that there are many middle-income countries around the world that already produce these killed whole cell bacterial-derived vaccines. The cholera vaccine and also the diphtheria, pertussis, tetanus, and pertussis killed whole cell vaccine. A UNICEF target price for the DPT vaccine, which all the kids get in the first year of life, is actually 25 cents a dose and being produced for that right now. One thing that we're using in our technology is a class of proteins called gram-negative autotransporters. These proteins are a single polypeptide chain. They're the way that gram-negative bacteria place many proteins into their outer membrane. Gram-negative bacteria have an inner membrane, a periplasmic space, and outer membrane. They need to place hundreds or thousands of different proteins into their outer membrane. Some of these are enzymes. Many of them are adhesins. If you're a gram-negative enteric trying to live within the GI tract of an animal, if you don't have a way to adhere onto the epithelial cells, you're not going to be there very long. These proteins are really interesting because a single polypeptide chain mediates the export of the protein. So they have an N-terminal signal domain, a C-terminal beta barrel domain that makes a little pore-like structure in the outer membrane of the bacteria, and then a central passenger protein domain that translocates through that pore to be exposed to the outside world. Now, subject to some limitations, you can replace native passenger protein coding sequence with sequence encoding recombinant proteins, including vaccine antigens. And so you can get this protein to put up to 200,000 recombinant vaccine antigens on the surface of every gram-negative bacteria once you've engineered them. So we synthesized a plasmid that has an antibiotic resistance marker and an origin of replication. It also has a gram-negative autotransporter expression cassette in it under the control of a rhamnose inducible promoter. We have type 2S restriction enzyme sites flanking our coding sequence enable us to do scarless cloning. And the parental version of this has an HA immunotag that we can use to check for surface expression. So when we transform that plasmid into E. coli, of course, no plasmid, no band, but then if we induce expression of the HA immunotag, we get a band. If we induce expression and then trypsinize the bacteria before loading on the gel, the band goes away. So this means that the HA immunotag is both on the surface of the protein, but it also indicates that translation and export to the surface are very tightly coupled, and essentially nothing is left in the cytoplasm, because if there was, you'd see a band here. That means that it's very helpful for the expression on the surfaces of the bacteria of highly hydrophobic proteins that might otherwise tend to aggregate or form insoluble inclusion bodies within the cell. So the vision here is that we identify a target antigen for vaccine production, synthesize the DNA, clone it into our plasmid, transform it into bacteria, induce expression, and have something that we can test in animals, of course, not in people, but over maybe just the course of 3 weeks. So we started doing this because we were working on HIV vaccines. And in the HIV vaccine field, there's an emerging consensus. There's the observation that some patients who are infected with HIV for a long time have sera that can neutralize many different HIVs. These are called broadly neutralizing sera. Then immunologists have made monoclonal antibodies from those patients' B cells, gotten broadly neutralizing monoclonal antibodies. And then structural biologists, including my friend and collaborator at the NIH Vaccine Research Center, Peter Kwong, who's a brilliant structural biologist and highly prominent in this field, have then mapped the sites on the viral envelope that bind those broadly neutralizing monoclonals. So then the molecular engineering problem becomes to try to make an immunogen that mimics the binding site of one of those broadly neutralizing monoclonal antibodies. So here's a figure from a review paper looking at the different classes of binding sites for the different broadly neutralizing monoclonal antibodies. And I'm going to highlight two. One is the fusion peptide, which after the envelope protein binds its receptor, undergoes a conformational change. The fusion peptide is the molecular machine that helps catalyze the fusion of the viral lipid bilayer envelope with the host cell lipid bilayer to enable entry into the host cell of the viral genetic package. And the C term, what's called for HIV, the membrane proximal external region, which is the stalk region that comes out of the viral lipid bilayer before it goes out to make the rest of the head of the envelope protein. These two are interesting engineering targets because they are simple linear peptides. Some of these other ones are highly conserved, but they have contributions to their sites from various different disparate parts of the molecule, making the engineering target much harder. So once that emperor was identified as the target for a class of broadly neutralizing monoclonals, the first thing, of course, people did was synthesize that peptide. Then they injected animals with that peptide. The animals made sera, but those sera were not neutralizing. So then the hypothesis was that absent the context of the rest of the native envelope protein, the emperor did not assume its correct tertiary and actually quaternary structure. So Peter Kwong's lab designed what they call scaffold emperors, or emperor-derived sequences that are expressed in a heterologous protein, computationally designed to fold into an emperor-like shape. So I was actually at the VRC a few years ago giving a talk on a different subject. And afterwards, I was talking to some of the folks there, and I was talking to Peter, and Peter told me about this problem. And I said to him, Peter, I think I have the solution for you, because we're working with these gram-negative autotransporters. Translation and export to the surface is very tightly coupled. if you give us the sequence for your Scaffold Emperor, we can clone that into our expression system. It'll go out on the surface before it has time or concentration to make an insoluble aggregate, and we can express your scaffolded emperor that way. So Peter kindly gave us the sequence. We synthesized the DNA, cloned it into our system, and tested it. I'll show you that in a few minutes. Now, it's interesting because there are actually not just one, but two classes of gram-negative autotransporters. There's the monomeric kind, and then there's a trimeric kind. And the trimeric kind has a beta barrel that's composed of 3 different molecules, and 3 passenger proteins translocate through that pore. That's very interesting because the structure of the trimeric autotransporter very closely resembles the structure of the stalk region of many different envelope viruses. Although influenza, HIV, and actually I don't have it here, but coronaviruses are totally different virus families. Nonetheless, they are all classified as having type 1 viral fusion proteins, in which you have a trimeric stalk that comes out of the lipid bilayer. So we hypothesized that if we could just use a recombinant version of this, in which a native emperor peptide was put out on the surface using this autotransporter expression cassette, we might be able to have a structure that closely resembled the HIV membrane proximal external region. So we made bacteria transformed with these plasmids, either expressing the Scaffold Emperor or the native trimeric emperor, did flow cytometry using a broadly neutralizing monoclonal directed against the membrane proximal region, and showed that we had binding in both cases. And then we went on to, this is just data that actually Wen Yuan in the Department of Medicine here collected for us, where we immunized mice intranasally with a killed wholesale bacterial vaccine made from the Scaffold Emperor sequence that Peter designed. And what you can see here is that as you look for neutralization, you get some at a low dilution, which dilutes out in most of these mice as you go to higher dilution, suggesting that it's specific. Of course, the mice immunized with control bacteria had no such response, and the neutralization assays done with the highly active anti-emperor monoclonal antibodies gave you a very impressive neutralization dilution response. So that was good, but we weren't getting quite as much neutralization as we wanted to get. So we had to ask ourselves, how can we make that better? So we took a step back and we thought for a moment, and we said, well, you know, we're putting these vaccine antigens on the surface of bacteria. The bacteria have hundreds or even thousands of other proteins on their surfaces. We, in our sort of self-absorbed way, know what we want the immune system to pay attention to, but the immune system doesn't have any idea about that. How does the immune system know to make an immune response against the one recombinant vaccine antigen that we're putting onto the surface of bacteria versus all the other ones? So we had a kind of simple idea, which is, let's just take a lot of those other proteins off the surface of the bacteria and see whether that makes the bacterial, killed wholesale bacterial vaccine expressing the antigen that we care about more antigenic, or at least more visible or accessible to the immune system. We also thought that if we start to take things away from gram-negative bacteria, we may also enhance the immunogenicity of the bacteria, because commensal bacteria, or even gram-negative bacteria, have evolved to try to not elicit a potent immune response, because if they did, they wouldn't be living in the GI tract for very long. So we thought if we start to take things away, we can disrupt or somehow expose more, make the bacteria more immunogenic overall. So how do we do that? Well, I have kind of a small lab, so I don't really have the personnel to make extensive deletions of bacteria. That's a multi-year project for a whole group. But it so happened that around the world, people had been asking a kind of fundamental question, which is, how many genes do you need to make a living bacteria? I think that's actually quite a very interesting question. Some people had taken a build-up approach where they started putting genes together and tried to design a bacteria from the ground up. Other people had taken a subtractive approach where they took a wild type bacteria and then started deleting genes, delete, and tried to see how many genes they could delete and still have a viable bacteria. So this group at the Tokyo Metropolitan University did a really elegant project in that regard, and they were actually able to delete up to 29.7% of the E. coli genome and still have a living bacteria. So those bacteria grow slowly with a doubling time about twice as long as a normal doubling time. And they have an altered morphology. They look ugly, but they're still alive. So we got this whole collection of genome-reduced bacteria from the group in Japan. And then...
Can I ask you a quick question?
Yes.
Sounds really exciting question, right? So two related questions. One is, how do you define that you still have the bacteria? I mean, what functional aspects does it have to preserve? And second is, you would expect different subsets of genes to be removed and still have the notion of a working bacteria. So I was just wondering, you must have some specifications to this question, right? But
right.
Very interesting.
So the definition is simply an operational definition that it's alive. So they deleted the genes and then they put it in medium, and could the bacteria grow or could the bacteria not grow?
Okay, so replication is the only operational definition in some way.
Yeah.
Okay.
Yeah, it's just, is it alive or is it dead? And they're still alive if you delete that 29.7% of the bacteria.
Interesting. Okay, thanks.
So they knew which genes they deleted, and then an undergraduate working in the lab, Saraming, no relation to XJ, did an analytical look at those genes and imputed their location and found that in the most genome-reduced bacteria, up to 200 of the genes that were deleted should encode proteins that were present on the surface of the bacteria. And then when we did a flow cytometric experiment looking at when we expressed the HA immunotag on the surface of those bacteria, found that the binding of an high affinity monoclonal antibody against that HA immunotag went up as the percent deletion went up, suggesting that indeed, as you got more and more deleted, the antigen became more, in a sense, visible to the immune system. And so we also tested to see whether we could express the HA immunotag in these different genome-reduced bacteria, and we could, and by the trypsinization study, in addition to this flow cytometric data, got indication that we had expression on the surface of the bacteria. I talked initially about the membrane proximal external region target for HIV vaccine development, but I also mentioned that there's another linear protein epitope, the HIV fusion peptide. And so we synthesized that, put it into our expression cassette, expressed it on the surface of the bacteria, and found that we could, testing with a broadly neutralizing anti-fusion peptide antibody, could detect binding to the surface of those bacteria. And again, we got much better binding when we used the highly deleted bacteria than the control bacteria. So we can put both the Emperor and the fusion peptide on the surface of the bacteria and at least get an immune response with one of the Emperor antigens that we used. So that was sort of where we were when COVID started. So then we thought, well, gee, if what we're trying to design is a technology that can produce vaccines appropriate for pandemic responses, now that we're in the start of a pandemic, maybe we should actually see if we can do anything for it. So if we look at the structure of the spike protein, which is this protein on the surface that you all have seen a million times on the surface of the SARS-CoV-2 virion, as I said before, it's a type 1 viral fusion protein that has a structure very reminiscent of all the other ones. And it has several different functional domains there. The receptor binding domain is as it sounds like, binds the receptor on the surface of the host cell and then subsequently triggers a conformational change in the envelope protein and spike protein that then leads to fusion of the two membranes. The receptor binding domain is highly immunodominant, and it is the target of most of the neutralizing antibodies that have been identified. And when you're vaccinated with one of the SARS-CoV-2 vaccines, you're vaccinated with this entire spike. And that's true whether that spike is being produced by your own muscle cells as the result of being vaccinated with an mRNA vaccine, or in the case of Johnson & Johnson, an adenovirus vector vaccine, or in the protein vaccine that Novavax is developing, a protein vaccine, and so on. It's the entire spike protein. Part of my job is being an infectious disease doctor. And one of the things we've learned in infectious diseases over and over and over again is that pathogens have evolved to elicit immune responses against a part of them that doesn't matter as much to them. So that's true for HIV. It's true for gonococcal pili. It's true for trypanosome variant surface glycoproteins. It's kind of a universal evolutionary truth for pathogens. But really also for the SARS-CoV-2 spike protein, we have these other two regions that we can look at, the fusion peptide and the stalk region, just like in HIV. So I think the lesson, despite the fact that these vaccines are incredibly helpful, the lesson is maybe if you want a very durable vaccine and a vaccine that's resistant to evolution or variants, maybe you don't want to strictly mimic the exposure that patients have to the native protein on the surface of the virion. Maybe you want to be a little bit more clever and use something that is hidden, but nevertheless can elicit neutralizing antibodies. Probably a lot of you are familiar with this. This is a diversity plot I pulled out from nextstrain.org. You can see different amounts of diversity over the course of the entire genome. And then if we blow up the spike protein, we can actually see here over the receptor binding domain, there's a lot of diversity. But over the fusion peptide, there's very little. So if you want an evolutionary resistant, maybe even a universal vaccine, wouldn't you rather be targeting this than trying to elicit an immune response against something that can easily be mutated? And I think what we know from the clinical behavior of the other human coronaviruses, in other words, there are 4 human coronaviruses that are responsible for about 30% of the common cold syndromes that people get. You'll make an immune response against those, but that immune response is not durable, or at least doesn't provide durable protection. You can be reinfected with exactly the same cold-producing coronavirus in a couple of years. So maybe that's a lesson for long-term coronavirus vaccine development. So if we look at all the different fusion peptides in all the SARS-CoV-2 sequences that have been published so far, it's 100% the same. And it's very similar among all the different human pathogenic or epidemic coronaviruses. And even from SARS-CoV-1, there were monoclonal antibodies that were highly protective and protective in a passive immunization experiment 10 years ago in a non-human primate passive immunization challenge that mapped to the fusion peptide region. So in addition to the fusion peptide, there also is the stalk region for SARS-CoV-2. And working with Barbie Ganser-Pernios and Owen Pernios here in the biophysics department, we designed 12 different candidate stalk antigens that we could clone into not the monomeric version of our expression cassette, but the trimeric version of our expression cassette. to test because it's a little bit difficult on a structural biology basis to know from first principles which of the different variants would be better, how much transmembrane domain, and so on to include to get the best antigen. And so when we transformed bacteria with those trimeric SARS-CoV-2 stalk region expressing plasmids, we saw that some actually did show better binding than others. In all cases, again, we're always seeing that the genome-reduced bacteria have better binding than the other ones. But we've had some that were good and some that were less good. And actually, we have these going into animals early next week before Thanksgiving.
Quick question regarding using the spike part of the or the stalk part of the spike protein. Are there just potentially physical limitations on how the antibody can bind there just because it's not presented near the surface, it's somewhere underneath the spike?
I think, first of all, it's important to remember that these things are molecules and they're moving around at a molecular level. And what we look at in artist representations or even models don't represent the dynamic interplay that you see there. So something is relatively hidden, but there's enough Brownian motion or jiggling and joggling to express it from time to time such that the antibody can interact with it. That's actually why it's, I think, from the point of evolution, is hidden, because the virus doesn't want somebody to make a good immune response against, say, the fusion peptide, because then that would represent a huge challenge to the continued survival of the virus. So they hide, the viruses hide those critical targets. So for HIV, same thing. Only a very tiny fraction of patients infected with HIV make anti-HIV fusion peptide antibodies. Nevertheless, those antibodies are extremely potent. So if you can train the immune system to make those things, even though it's hidden most of the time, it's exposed enough of the time to enable those antibodies to neutralize the virus. Does that make sense?
Yes, thanks. That's great.
Okay, so here's SARS, MERS, the 4 human cold viruses. It's interesting, SARS-CoV-2 is a beta coronavirus. 2 of the cold viruses are betas and two are alphas. But when we were thinking of how to test this in animals, we noticed that there's a pig alpha coronavirus, porcine epidemic diarrhea virus, PEDV, that causes severe watery diarrhea in pigs. And my friend and collaborator, formerly your co-faculty member at Virginia Tech, XJ Meng, who is one of the world's eminent animal virologists and actually, one of the nicest scientists and best collaborators I've ever worked with, studies this. So we approached XJ and said, look, how about, would you be interested in working together with this? Because really, the cool thing about this is that at Tech, with their ABSL3 lab, they can test vaccines to prevent infection in the pig using the native host with its own pathogenic virus. So XJ said he would be super happy to do that. PEDV was a huge problem, really a pig pandemic. Pigs are a great model, readily available, and really a great opportunity to study the vaccine development. So we made vaccines against both the SARS-CoV-2 fusion peptide and the PEDV fusion peptide, showed that we could express these in the bacteria and that we got binding of anti-fusion peptide antibodies to both of these. And then these are XJ's lab members vaccinating these cute pigs over in Blacksburg. And then this is the structure of the study that we did. We tested 7 pigs and vaccinated them either with the SARS-CoV-2 or with the porcine epidemic diarrhea virus vaccine, and then tested them. And what we found was that we vaccinated and then boosted and then challenged the pigs, and we could get a little bit of humoral immune response after a single boost. But then after the challenge, we got a very potent anamnestic response, and also by interferon gamma, some evidence that there was a cell-mediated immune response as well. And we saw differences in the amount of virus that were in some of the key tissues, GI tissues of the pigs, and also looked at different measures of disease in the pigs, intestinal content score and villus length to crypt depth score. But really the most interesting thing, I think, was that we could show that we had a strong, significant clinical protective response in the pigs when we challenged them with the PEDV. One is normal for both the diarrhea and the body condition. So this means that we can protect the pigs against PEDV when we vaccinate it with the PEDV vaccine, but we can also vaccinate with the SARS-CoV-2 vaccine, a beta coronavirus, and get cross protection against the PEDV, suggesting that we actually might have a pathway to developing a universal coronavirus. So the conclusion here is we can express the fusion peptide and EMPER. We can express vaccine antigens on the surface of genome-reduced bacteria. They appear to be more visible to the immune system, and we can protect pigs against clinical disease from either the alpha or beta coronavirus fusion peptide vaccines. And we can do other potential vaccine targets as well. So we're not stopping there, but we're trying to optimize the immunogenicity by using additional targets and additional genome-reduced bacteria, working with our collaborators now at the International Vaccine Institute, looking at different antigens. So not just a monomer of the fusion peptide, but multimers of the fusion peptide, and using better bacteria and better cells. And then mini cells, which I'll show you some data on. The bacteria that we use, the genome-reduced bacteria that we use, were really opportunistic, but we're working with or in discussions with some bacterial engineering companies to delete even more genes encoding proteins on the surface of the bacteria. And maybe you doing that for a pathogenic bacteria, namely Salmonella, so we would get an anti-pathogen immune response as a background, and also enhance the bacteria in some other ways. We also want to, as I mentioned before, make a multimeric version of the fusion peptide, which we think will be much more effective. And this shows you some data there. If we can make a 1-mer, 3-mer, 5-mer, and then also adding some extra aspartates to the end, which seems to enhance production of the 5-mer. And this is actually going into mice next week as well. And then working with Mark Kester here at UVA to make mini cells from these genome-reduced bacteria. If you delete the MinC and MinD genes from the bacteria, you don't get the division plate forming in the middle of the bacteria. You have division forming at the very end of the bacteria, resulting in the budding off of a small mini cell. The mini cell then has a much higher ratio of surface to cytoplasm, which we think may concentrate the antigen and also maybe more effectively interact with the reticuloendothelial system, be more able to be transported across mucosal barriers and so on. And also the autotransporter cassette that we're using concentrates at the bacterial pole so as to further enhance concentration of the antigen. This is just an electron micrograph of mini cells made from an intact bacteria. And this is a figure from an old paper showing that the autotransporter that we're using is concentrated at the pole of the bacteria, which should help in the production of a more concentrated immunogen. So we make a MinC, MinD mutant, and we get a little mini cell from the bacteria that has the protein of interest on its surface. We can make mini cells from both the wild type and the genome-reduced bacteria, whether it expresses proteins in the cytoplasm or on the surface. And then when we do image flow cytometry, we can in fact see that there's concentration of the antigen, both in the cytoplasm and on the surface, about 2.5-fold versus the parental cells. So we think that'll be a much more immunogenic bacterial-derived vaccine. So the other thing we want to do is work for a non-parenteral vaccine. So the killed wholesale cholera vaccine is administered orally, so it's reasonable to think that we should be able to do that. We're going to be testing different doses, schedules, and routes in an attempt to further optimize that. And I initially talked about the need to do this in a globally appropriate fashion. So this is an image I pulled off the web, a 1,500-liter bioreactor at a company called Incepta Vaccine. It's Bangladesh's indigenous vaccine production company. Bangladesh is a country of about 160 million people, a kind of lowish middle-income country at this point. This 1,500-liter fermenter supplies all the DPT vaccine for the entire country of Bangladesh, as well as making the killed wholesale cholera vaccine at the appropriate prices that I mentioned. So this is just one of dozens of examples of indigenous bacterial vaccine production companies available around the world. So once we have our bacteria and once we have the processes developed and optimized, which our collaborators at IVI are doing and are adept at doing, IVI actually transferred their technology to this company. We should be able to deploy any vaccines that we come up with around the world very quickly and produce it at a very low price. Beyond vaccines for infectious diseases, this may be helpful for making custom polyclonal and monoclonal antibodies and perhaps custom cancer chemotherapy targeting either known tumor-specific antigens or tumor-specific antigens identified in a particular tumor via RNA-seq or proteomics. And because we can take just 3 weeks to go from target to testable vaccine, this may be very helpful in the cancer immunotherapy dimension. So I put this slide up here. So how does this enter? Maybe it's been percolating around in your minds, but your focus is on pandemic research and emergence planning, and I'm going to highlight this here, response. So once you guys detect, forecast a particular outbreak, then what do you do next? Well, if you can port that over into a bioinformatics, molecular virology, immunology activity, you can identify antigens on the pathogen surface, perhaps informed by knowledge of antigens that are previously known to be targeted by neutralizing antibodies of that pathogen class. We can put it into our system and then make a testable vaccine. So here you go. This is you guys finding and IDing and selecting a particular antigen. And I think once you know the identity of the pathogen, it's the cause of the outbreak, you could do this very quickly. Feed it into this, we synthesize the DNA for that, and then have something that we can test. So I hope that it maybe was what you were thinking about, as I was talking, but in any case, I think it's really an area that would be very ripe for collaboration. So anyway, so I think the takeaways, we have this new synthetic biology platform that I think is quite disruptive, rapidly producing vaccines for infectious diseases, but also for other applications at a less than $1 a dose in existing factories around the world, including low and middle income countries. We've already started this with HIV, SARS-CoV-2, and PEDV, and we're working to optimize that and get to a point where we can start doing clinical testing. So here's my acknowledgement slides. We've been very fortunate to have some extra funding. XJ's been great, and these are the folks in my lab, and Mark Kester for the Minicell and the WHO International Vaccine Institute going forward to develop, deploy, and optimize this around the world. So I'll stop there and be delighted to answer any questions you might have.
Thank you very much, Steve. This is an excellent talk, absolutely fascinating.
So really a wonderful talk. I really found it really interesting. In this last part that you said, again, I didn't understand a lot of it, but if you find a sequence and put it in, are you also able to look at possible mutations that might happen and incorporate that as well?
Yes, absolutely. I mean, the beauty of this is it's synthetic biology based. So it's, for 15 cents a base or less, we can synthesize anything we want to. So if there is a variant that we need to cover, we can synthesize that and get the DNA in 5 to 7 days, put it into our system, and then do that. If you're also talking about pandemic response, there's a limited number of viruses in this world. So I think you could even kind of pre-consider different classes or different families or different genuses of viruses that have pandemic potential, pre-identify vulnerable areas in those viruses that might be suitable for this, and then look for conserved sequences in those vulnerable areas so that you'd have something ready to go if you detected something.
Thanks.
Steve, just to follow up, more not a question, but a possible place to even work further. Andrew here and Mandy in her past life used to do work on synthetic biology, and one could imagine computer programs that could take a basic genome for a virus, but look at possible variations, such as the one Anil talked about, right? We don't know how it's going to mutate, but we could come up with a set, possibly through understanding of how these things go and what stable systems it can produce. And perhaps that's one way we could study, but I'd let Andrew and Mandy also talk a little bit, because Mandy actually worked on problems like this in the past.
You know, one question I had along those lines, and this may have been covered, I may have missed it, but is there the ability in the system to sort of multiplex the targets and come up with a polyclonal stimulus in the delivered sort of package?
Yeah, so the answer to that is absolutely. It could be done two ways. You could have multiple different bacteria, each expressing a different antigen, which is sort of the easiest way to do it. The other way is that when I was talking here about making a multimeric version of the fusion peptide vaccine, these can be homomultimers or these can be heteromultimers. And actually, this is in a grant application that we put in as a program project with XJ and some other folks at Tech. So if we're looking to make a universal coronavirus vaccine, one of these fusion peptide sequences could be from SARS-CoV-2, one could be from SARS-CoV-1, one could be from MERS, one could be et cetera, maybe even a couple of bat coronaviruses, so that you'd have antigenic representation from all of the likely pathogenic or pandemic capable viruses. But absolutely.
Right. Thank you. Thanks for the wonderfully interesting talk. That was amazing.
Thank you.
Yeah, Steve, I mean, first of all, I really appreciate you coming and talking with us. We've been wanting to do this for a while, so thanks very much. And I think P2P really offers us a chance to now do this work together. One question I had, and then in close, is on a modeling side, if we design these vaccines and we have some understanding of its response, in terms of reducing the chance of either getting sick or reducing how hospitalization maybe, or the level of infectivity, then one could actually couple. So you design this vaccine, perhaps there's some understanding of the level of things it can bring down, and then we could study the epidemiology of it if you were to administer these vaccines down. So there's a part where we can couple with you on the front side, but there's a part that we could potentially couple with you on the other side, where we can look at the consequences of the vaccines you make, because there might be price versus efficacy points. There might be variants of it that can help. Maybe certain populations might have certain characteristics where the vaccines might be better. I quite don't know, but I would think that would be the case.
Yeah.
And we could study that.
Absolutely. No, completely.
Great. This is great. I mean, I really enjoyed it. If there are any last set of questions from the group, else we'll thank Steve again and hopefully continue the collaboration.
Okay.
Hopefully we can do it in person, Steve. Next time we'll invite you again, possibly in spring, if you have time and energy to talk to all of us. And maybe we do it in person and have coffee.
Okay.
With us in the new facilities that we have.
Okay, love to do that. Thanks so much for the invitation.
Thank you very much. Take care.
Bye.
Yeah. Thanks.
Thanks, everybody.
Bye.